Hey guys, welcome back. This is Lei. Previously, I showed you how a single gene can encode multiple isoforms through alternative splicing. In this video, I'm going to show you how to quantify the expression of these isoforms. The tool we're going to use is the Linux command line tool called Salmon. Salmon is a super powerful quantification tool that quantifies gene expression at both isoform level and the gene level. Compared to our previous star and feature counts combination, Salmon is extremely efficient. It can finish the quantification in under 10 minutes. So if your goal is just to quantify gene expression, Salmon is the way to go. Now let's set up the tool. Here we use the RNA-seq environment we created before and install Salmon using conda. And if you haven't installed trim-galore, install it now. And check the installation using Salmon version command. And don't forget to download the Salmon index from RefGenie. Make sure you specify the correct species you're working on. Just download everything in the red box and put all the files in a properly named folder. Once set up, we can start our quantification process in 2 steps. Step 1. Trim the adapters using trim-galore as we did before. Here we have 2 FastQ files, R1 and R2, for each sample. Make sure to store them in a properly named output folder over here. To set up Salmon, we do this, Salmon quant-i to specify the Salmon index we just downloaded, -LA to set up the library type, basically the orientation of the reads, -1 the first FastQ file, -2 the second FastQ file. The validate mappings option makes the mapping more accurate. Just leave it like this. -P means how many CPUs you want to use. Here I use 8. -O the output folder over here. So the results are stored in a tab delimited table named quant.sf in our output folder in the following format. First column, the ID of the transcript. Length, the length of the transcript. Effective length, you don't have to know. This is the length that the algorithm used to calculate the quantification. TPM, transcript per million. This is the normalized expression value. Num reads, this is raw counts. They're not whole numbers because the algorithm is different from the previous one we used. But this is perfectly fine. So if you want to do differential expression analysis using the counts over here. So here's the problem. So we have transcript IDs, which is not much informative. So we have to quantify and annotate the transcripts. So next step, annotate the transcript IDs. We can do this in R. So actually, the code looks complex, but the logic behind it is very simple. We just need to create an annotation table in this format. The first column is transcript ID. The second column is the transcript name. So all this information is stored in our GTF file, the genomic annotation file. So when you download the Salmon index, there is a GTF file already in it. So just read it in using fread. And on the third column, we only need transcript. And then all the information is stored in the 9th column. Basically the transcript ID and the transcript name. So the whole code, the piece of code, is to transform the GTF table into this format, only 2 columns. So this is logic. You can try it on your own. So next, we're reading our quant.sf, the quantification table, and remove the version number, this dot something here in our original file, the .2.3, this version number of the transcripts. We don't need them for annotation, so we remove them, and we map the transcript IDs in our annotation table with our IDs here. So we add a new column called transcript name. It has all the names of each transcript. You can rearrange the orders of the columns to make it easier to read. So if you are having like multiple samples, there's an easier way to combine them and annotate all of them in one go. Here, we use another package called tximport and trim-galore to combine all the quantification tables, load the packages, install them in this way, and then we list all of our quantification tables, gene expression matrices, in a list, and then we'll read all the files using one function called tximport from the same named package, and specify the options like Salmon and TPM. Read all of them. So this function read all. I have 4 samples, so it reads in all 4 gene expression matrices and store them into an object named txi. Of course, you can change the names. And in this txi object, there are multiple tables. So you can access these tables using the $. So just type $, after the object name, and click on tab on your keyboard to see what's inside. Now we want the counts. So we access the counts in the txi object. And then we remove all the version numbers after the transcript ID. And then we do the same thing to map the transcript names to our IDs and rearrange the columns. And we get this table, the merged counts table. So first column is transcript ID, second column is transcript names, and all other columns are the counts of the transcripts. So this is the transcript expression matrix you can use for the following differential expression analysis. So if you want TPM, so just go to txi object, type $, and type abundance, because there's another table called abundance. This is a table with all the TPMs. So if you want TPMs, access TPM this way. And you can map the transcript names in the same way and rearrange columns as we did before, and save the TPM table on your computer using the fwrite function. So these are the TPMs here. So once we have the count table and the TPM table, you can perform a differential expression analysis as we shown in other tutorials. Same way, just use count table, because most of the commonly used differential expression packages only accept counts. We can also quantify gene expression at gene level, but here we need to make a gene symbol and the transcript ID annotation table. So we use a different function in the trim-galore package. Here we use the import function to import the GTF file, and we extract the isoform ID and gene name to make an annotation table like this one. First column, transcript ID, second column, gene symbol. And we use the same function from the txi import here to read in all the files, the transcript level expression files, and then we specify tx2 gene option. We put our annotation table here, and then the gene level quantification is done. You can just access the counts using the $ counts and TPM from the abundance table and save the results to your computer. The resulted gene expression matrix is in this format. Now they're all in the gene level expression. So this is how we use Salmon to quantify transcript level and gene level expression. And now you can go to my website and copy the code and try it on your own. And in the next video, I'm going to show you how to perform alternate splicing analysis at isoform level.